Okay, good afternoon. Welcome to the Distinguished University Professorship Lecture Series. I'm Lori McCauley, Provost and Executive Vice President for Academic Affairs. And let me just say, this is like one of my all-time favorite events on our campus. Those of you in the audience, you are really in for a treat. We have three colleagues who I just think the world of all of them, and we're going to be really in for a great session. Each year, this series stands as a reminder of why universities matter: to create, to preserve, and to share knowledge in ways that shape lives and expand our collective imagination. This event is not only a celebration of scholarly accomplishment, but also a chance to glimpse the frontiers of research and teaching through the lenses of some of our most distinguished colleagues. First, let me thank Mike Solomon, the Bernard J. Lucci Professor of Chemical Engineering, our Vice Provost for Academic Affairs Graduate Studies, and Dean of the Rackham Graduate School, who will be my partner and will be moderating the Q&A. Thank you, Mike. I also am grateful for the many staff and organizers who make this all possible, in particular our colleagues from OUD, our events team. Thank you. And of course, to our speakers for sharing your work and your insights. These scholars represent the breadth of inquiry at Michigan, spanning history and culture, biosciences, and medical imaging. At first glance, you might think these disciplines may seem to stand worlds apart. Yet what unites them is unmistakable: a drive to pursue questions with unflagging curiosity and to illuminate new knowledge. Equally important, each of them has carried their insights beyond their research through teaching, through mentoring, and innovation that have left a lasting impact on our institution, on their students, colleagues, and society at large. They've launched ideas, built programs, spun off companies, and nurtured generations of scholars who continue their legacies. It's my privilege to start and introduce Deborah Moore, the Jonathan Friedman Distinguished University Professor of History and Judaic Studies in the College of Literature, Science, and the Arts. Professor Moore is a world-renowned scholar of American Jewish history and Jewish women's history. Since joining U of M in 2005, she has transformed the Frankel Center for Judaic Studies into an internationally respected entity. Her scholarship has explored themes that others have overlooked. She wrote about America when Jewish scholars focused on Europe. She wrote about women when the emphasis was on men, and Jewish identity when assimilation was often assumed. She's the recipient of 3 National Jewish Book Awards and the Lee Max Friedman Award Medal, among many others. Her lecture, Vernacular Religion and Robert Frank's The Americans, investigates the work of the photographer Robert Frank and reveals how his images captured the diverse expressions of vernacular religion across the United States. Through this lens, she offers a Jewish interpretation of American culture that's as insightful as it is timely. Please join me in welcoming Professor Deborah Moore.
Thank you very much, Provost McCauley, and for when we get questions, also your ability to handle those. I'm going to start by saying that I will be showing some photographs, and the screen here doesn't work very well in terms of letting you see the photographs well. So once the photographs come on, I suggest you look to the sides, okay? In April 1955, Robert Frank a Jewish photographer applied to the Guggenheim Foundation for a fellowship to, and I quote, photograph freely throughout the United States using the miniature camera exclusively. That is, a small format 35 millimeter handheld camera. Frank was an outsider to America, and as a photographer, he was also a professional stranger. Born in Switzerland in 1924 to a German Jewish father who was declared stateless in 1935 after Hitler came to power, and a Swiss Jewish mother, he grew up in a politically precarious bourgeois household. Provisionally protected from the Holocaust by Swiss neutrality during World War II, Frank recalled experiencing anti-Semitism as an organic part of life. As he later said, hearing Hitler on the radio, talking, threatening, cursing the Jews, it's forever in your mind, like a smell, the voice of that man. After graduating high school, Frank refused to enter his father's business. Instead, he learned photography during the war years through apprenticeship and employment. He gained Swiss citizenship in 1945, although it was denied to his father, and at the first opportunity, wriggled free from the Swiss cocoon that had kept him alive. Almost as soon as the war ended, he left Switzerland for Paris, and then in March 1947, emigrated to New York City. Frank came as a footloose, ambitious young man. He arrived in the United States without command of idiomatic English and without immediate prospects of employment. But New York City's inconceivable tempo impressed him. He penned his first impressions to his parents. Quote, "Never before have I experienced so much in 1 week as here. I feel as if I'm in a film," he wrote. "This is really a free country." He continued, "There is only one thing you should not do: criticize anything." By the time he applied for the Guggenheim Fellowship, the United States was just recovering from the throes of McCarthyism, the Cold War was raging, and Americans were flocking to the suburbs. Frank, too, had changed. He was 30, married, a father, and despite rejection from membership in the photojournalist cooperative Magnum, his photographs had achieved recognition in both publications and exhibits. In his Guggenheim application, edited and rewritten by American photographer Walter Evans, Frank admitted, "The photographing of America is a large order. Read it all literally, the phrase would be an absurdity. What I have in mind then," he explained, "is observation and record of what one naturalized American finds to see in the United States that signifies the kind of civilization born here and spreading elsewhere." Using Evans' language, Frank proposed not a carefully planned trip, but a spontaneous record of a man seeing this country for the first time. Frank received the Guggenheim Fellowship to spend a year photographing Americans. He subsequently received a second year of funding. He initially left his home and family in New York City, driving first to the Midwest, and in fact, he visited Ann Arbor and took some photographs that made it into the book. And then south before heading to the West Coast, returning through the Northwest. Frank shot many thousands of photographs. At the end of his mammoth 2-year project, he selected only one out of every 300 shots. 83 in all for his book, The Americans. It was published in France in 1958. With some difficulty, Frank then found a publisher in the US. This edition of The Americans appeared in 1959 with a foreword by the beat writer Jack Kerouac. The book ignited storms of controversy. However, over the years, The Americans has come to be widely regarded as one of the great photographic books of the 20th century. And this is Ann Arbor. One of the things that Frank saw in America was vernacular religion. As the religious studies scholar Leonard Primiano observes, vernacular religion is, by definition, religion as it is lived, as human beings encounter, understand, interpret, and practice it. Primiano explains, vernacular religion is more than a purely conceptual term, a methodological abstraction without any practical utility. Full understanding of this term establishes a methodological foundation from which the vernacular study of religion will grow as a method of practice, a way of doing ethnography that has not been considered before. The anthropologist Karen Nakamura, while admitting that ethnographers consider the category of photoethnographers a contentious one, includes Robert Frank in her list of photoethnographic artists. Significantly, some professional photographers were open to the conceit of photographic practice as ethnography. Looking at Robert Frank's photographs of American vernacular religion connects several modes of interpretation. Frank presented himself in his application as a naturalized American, implicitly an outsider. The term, often used with reference to Jewish subject position, is contextually complicated. To what extent did various configurations of outsider status characterize Frank? His understandings and sentiments, his work, his reception, and reception of his work. A dependent relative status, outsider, refers to that which is external to some other thing or condition, which, at least figuratively, is inside. We commonly assume that an outsider is less knowledgeable and less secure relative to those on the inside. Even when inside, he is outside, looking in, an aspirant, foreigner, expatriate, exile, or refugee. With respect to his outsider status, he may be perceived as quaint, mysterious, resplendent, feeble, or bigger than life. From Alexis de Tocqueville to Georg Simmel's Urban Stranger, to Hannah Arendt's Conscious Pariah, to Yuri Slezkin's Mercurian, the outsider figure has also been praised as peculiarly insightful, even objective in some respects. In a book about ethnography, Michael Agar notes that people may be drawn toward careers as professional strangers because they feel alienated from their native culture, or because they grew up loving cultural variety. In either case, to broaden his point, they may find in ethnography, and I quote, a professional role to justify their move from one way of life to another. His comments describe well the dynamics that pulled many young Jews into photography. The camera became a cultural and social passport. This uncanny tool could transform the familiar and foreign alike. Robert Frank was arrested in McGehee, Arkansas, while he was on the road taking photographs. The unnerving experience drove home his insecurity as an outsider, an unattached foreign Jewish photographer in the United States. Imprisoned for 40 hours in a local jail, Frank was questioned by police officers about his Jewish background because he had letters from the Guggenheim Foundation and other individuals with Jewish names among his possessions. Frank had attracted police attention through his New York license plate, shabby dress, and his need of a shave, haircut, and bath. The police noticed his accented English and feared, 2 years after the execution of Julius and Ethel Rosenberg for conspiracy to commit espionage, that he too was a Jewish communist spy. The first picture of vernacular religion in the Americans, taken in Hollywood, California, appears on page 37. It portrays a young woman in the Ranch Market, one of the many diners and lunch counters that attracted Frank's attention. He photographs her directly below an oval sign of a smiling Santa. Santa is looking to his left, She is looking to her right. Above Santa's head is the greeting, Merry Christmas. Signs for hot dogs and steak sandwiches flank Santa, leaning against shiny aluminum reflecting light. One promises absolutely no fillers in the beef burger, guaranteed by the owner. In the US in the mid-1950s, popular culture championed Christmas as an American holiday. It pervaded all sorts of quotidian spaces, like luncheonettes and diners. One can almost hear the Christmas music playing in the background. The contrast with Christmas celebrations in Switzerland probably struck Frank forcefully. 2 pages later appears a photograph that Frank labeled Yom Kippur, East River, New York City. Frank obviously wanted to include Jews among the Americans, and he chose to portray them through vernacular religious practices. The photo pictures Orthodox Jewish men and boys from behind, their heads covered in hats or yarmulkes, gazing down, perhaps at a prayer book, perhaps at the river below them. If one reads from right to left, as one does in Hebrew or Yiddish, the young boy in the lower corner invites one's eye to travel up past the young man wearing a neat pinstriped gray jacket to the dark mass of men dressed in black. Frank grew up in a non-observant home, albeit one that recognized the annual holy day of atonement, Yom Kippur. So he mislabeled the photo. It is not Yom Kippur, but the Jewish New Year of Rosh Hashanah. The men and boys have gathered, probably by the barely visible Williamsburg Bridge that spans the East River, connecting the Brooklyn Jewish neighborhood of Williamsburg with Manhattan's Lower East Side. For Tashlich, this vernacular religious practice of casting away one's sins was initiated by pious Jewish women in Eastern Europe before being adopted by men. It takes place in the afternoon of the holiday when Jews gather by flowing water to say prayers and throw bits of bread into the water to wash away their sins for a clean start to a new year. The photograph closes the first section of The Americans. Not until another 24 pages does the next photograph of vernacular religion appear. It shows a Jehovah's Witness standing in front of a massive wall in front of an etched cross dividing the building's stones in Los Angeles. He holds a copy of Awake, the Witness's magazine, in his left hand. Unlike the Orthodox Jews, this man knows that he is being photographed. He leans forward slightly, looking directly at Frank and his camera, his right hand in his pocket, his briefcase at his feet. He is witnessing to his faith, to Frank, and through the photograph, to anyone who looks at it. Jehovah's Witnesses were an indigenous American religious movement, albeit one that had spread to Europe by the 1950s. The movement began in the 19th century and developed distinctive theological doctrines and religious practices. In the 1950s, witnesses were known most often for rejecting military service and blood transfusions. An active evangelical faith, Jehovah's Witnesses seek to convince people to engage in Bible study, a continuous practice dating from the movement's founding. This photograph, taken in Santa Fe, New Mexico, may not have captured Frank's eye because of its religious implications, although the large "Save" sign certainly intimates something more than cheap gas. Like many Europeans, Frank found American car culture fascinating. The gas pumps here seem like so many robo-icons ready and waiting to be saved. It helps to see the religious vernacular and humor in the previous photo by juxtaposing it with 2 more explicitly religious vernacular photographs, 10 pages later, that follow each other. The first, Mississippi River, Baton Rouge, Louisiana, shows a white-robed and hatted black man. He faces the river, kneeling on discarded cardboard, and holds a white cross in his right hand and a tambourine and Bible in his left. Frank pictured him in the middle of the photo, with the river a long triangle of light above him. Frank took the photo shortly after his unnerving experience of being jailed for 2 days and questioned intensively for 4 hours in Arkansas. The black preacher in the photograph was famous for walking the length of the Mississippi River. As he knelt to pray in the fading light, he shone forth like a beacon from the drabness of the scruffy terrain. Photography historian Sarah Greenow considers this photograph one of Frank's most compelling images, one that, quote, speaks not only of the depth of the man's belief, but also of the inexplicable nature of all faith. The photo on the next page portrays a statue of a priest standing, right arm raising a cross in a posture of blessing the street before him. Facing him is an auto repair shop, and next to it, a gas station and a huge parking lot filled with cars. Frank captioned the photograph, as you can read, St. Francis Gas Station and City Hall, Los Angeles. A Catholic saint, Francis was known for his devotion to the poor and founding the Holy Order of Franciscans. Without Frank's identification, it would be hard to recognize City Hall looming in the distance, behind the auto shop. Together with the gas station and parking lot, these might be seen as icons of Los Angeles, the city of angels, religious faith, specifically Catholicism, car culture, and American government. Frank often photographed American flags. Here, City Hall stands in for political faith. Beyond City Hall, the sun glows faintly, muted by smog behind barely discernible mountains. Frank's placement of the two photos invites reading them together, the black minister and the statue of St. Francis, showing how Frank portrayed an integrated vision of American vernacular religion, one that included blacks as well as whites. Frank later recalled that he was attracted to the saint because someone had placed a small vase of flowers at his feet. The personal tenderness of the flowers in such an concrete, impersonal streetscape touched him. The possibility of life after death seems to emanate from Frank's lonesome photograph of 3 crosses by a highway, US 91 in Idaho, marking deaths in an auto accident, illuminated by a shaft of redemptive sunshine. The American vernacular religious practice of commemorating highway deaths with crosses served as a grim reminder of the growing numbers of fatalities on the road. It brought consciousness of death closer, a potentially daily reminder of the fragility of life and the dangers of driving. Frank placed his last photograph of American vernacular religion near the end of the book. It comes just before the photo of Ann Arbor. This photograph explicitly combines car culture with religious practice and American political faith. Perhaps fittingly, it is an almost abstract image of a car, albeit one with several compelling, clear messages. Christ came to save sinners, attached to the license plate that proclaims Land of Lincoln, and taped onto the back window, Christ died for our sins. Bright lights reflects off the car, revealing shadows of metal fire escapes, stairs possibly to heaven. Although these 8 photographs all speak to Frank's fascination with American vernacular religion, there is one photograph that most likely did not express his engagement with American vernacular religion, but that can be read in that light. He took it on Beaufort, South Carolina, a town on one of the Sea Islands, largely populated by Black Americans who spoke a distinctive Gullah dialect. In the 1950s, the island still retained many mores associated with African Americans. The photograph portrays a woman sitting on a chair in a field. One arm holds the chair's back, and the other is akimbo on her hip. She looks off to her left, a broad smile on her face. Frank's photograph places her in the center of the scene. Behind her, the sun sinks into the horizon, lighting a utility pole shaped like a cross. Her dignity and joyful presence pervade the countryside. Looking back on his project, Frank later wrote that he had worked himself into a state of grace. Looking at his photographs today, I think we can agree and say amen.
Thank you very much for your lecture, Professor Dashmore. Thank you for coming back up. I was going to invite you back up if you're willing to take a few questions from the audience.
Oh, yeah.
We have microphones that will be brought to you. Please raise your hand or indicate otherwise if you'd like to offer a question. And as you're thinking of those questions, let me lead with one. You talked about an outsider looking, the perspective of an outsider looking in. But you also mentioned the storm of controversy that accompanied the publishing of the book. Could you talk a little bit then about the inside reaction to that view of themselves, please?
Okay, so a lot of Americans, including photo, critics and stuff like that, hated the book. They wanted to know why hadn't he taken pictures of happy people, of people smiling? Why were there so many pictures of Black Americans? Why, did everybody sort of seem angry in the book? It's not true, but you don't have people smiling. Where were the pictures of the beautiful suburbs that had just been built? He didn't take any of those. So it was one sort of critique after another. The book begins with a huge photograph of an American flag, and each of the 4 sections features the American flag. And the opening one is taken at a parade in New Jersey, and the flag is so big it blocks the people in the window who are out looking at the parade, and it's sort of, it's Frank's commentary on America. So you get flags in a variety of places. There's another flag photograph of a Navy recruiting office. You don't see the guy, you see his feet up there, and the shoe is sort of worn down, and the flag, and so he was making comments, critical comments about America, which I think, if you're looking at, as I was, at the vernacular religion material, you're getting a slightly different view, because he was really interested and engaged with this element of American society. Yeah.
Thank you. We have a question here. The microphone will come.
Thank you so much, Deborah. What a pleasure to have a chance to engage with your work here in this context. You know that I share an obsession with American photography and Jewish Americans in photography with you. And as you were working through Frank's images, it occurred to me to ask, to what extent did photography itself, maybe even beginning in the 30s with the New York Photo League, but to what extent did photography itself become a kind of vernacular religion for the Jewish Americans you're interested in?
Oh, that's really an interesting question. You always ask such good questions, Sarah. Okay, when you're a photographer, right, you carry your camera with you all the time. We have one in the audience here. It does become a way of experiencing where you are. It involves pausing and thinking about, do you want to take that photograph? If we were to think about it as a kind of vernacular religion, which I have not done before this question, I would say part of it then means that you're going to end up sharing it, that it's not just a lonesome activity, but it involves connecting with other people, and that's really possible, that especially in these years, starting in the 30s through the 40s, 50s, 60s and stuff. Yeah, this was a very, very powerful way and mode of existence. It certainly carried Frank out of Switzerland, to remaking himself in America. So, yeah.
Thank you for that. Please join me in thanking our distinguished university professor.
Thank you so much, Professor Dashmore, for that really captivating study of Frank's work. We really appreciate it. Okay, it's now my pleasure to introduce Roger D. Cone, the Mary Sue Coleman Director of the Life Sciences Institute and the Tadetaka Yamada Distinguished Professor of Molecular and Integrative Physiology in the Medical School. Dr. Cone is a global leader in obesity and metabolic disease research. His landmark discovery of receptor genes that regulate energy balance by the brain opened pathways in therapeutic advances for genetic obesity syndromes. Beyond his research, he's been a transformative leader at the university, guiding the Life Sciences Institute, directing the Biosciences Initiative, and championing innovative funding models to support promising faculty. He holds 20 U.S. patents. He's co-founded 3 biotech companies and has mentored dozens of rising stars in biomedical research. This evening, he will deliver his lecture, How the Brain Regulates Body Weight. In it, he will discuss how our neural architecture shapes one of the most pressing health challenges of our time, and how science continues to open doors toward new solutions. Professor Cone.
Thank you very much. Thank you very much for this honor. If I could have the first slide. Oh, I do that. Okay, there we go. So one of the fun things about this honor is that you get to name the professorship. And for me, this was easy. Tadetaka, or Tachi Yamada, was the luminary who I wanted to honor with this. He was a fantastic scientist who discovered many things about GI physiology and neuropeptide signaling. And he was chair of gastroenterology here, went on to be chair of medicine, and then had a very distinguished career bringing new drugs and vaccines to market. But I had 2 personal connections with him that were very meaningful. First of all, when I began my career in Oregon, he and I were competitors, and he worked in the same field I'm going to tell you about today. And then later when I came to Michigan, and he continued to serve the University of Michigan even after leaving, when I came to Michigan, I invited him to be on an external review panel for the Biosciences Initiative. And he graciously did so and gave us fantastic advice that really helped the initiative. So that's the origin of the naming of the professorship. So biological research and research in many areas of science is a team sport. Really, the professor is just kind of like a quarterback. It's the team, the team, the team, and I've been incredibly fortunate over the years to have amazing teams of students, staff, and trainees in my lab, not only from all over the United States, but from all over the world. And the diversity of talent and perspective that these folks have brought to my, it's really been essential to my discovery journey. And I also have to say, part of the team, and today is Research Administrators Day, and part of the team also includes fabulous universities like the University of Michigan, incredible staff like the staff at the LSI who provide the facilities and the resources and the financial management and everything you need to do research. So I begin with that. But what I want to tell you about today is what we've learned about how the brain regulates body weight. Now, it doesn't matter whether you're that super lean, 2-time gold medal winning marathon runner, Eliud Kipchoge in the middle, or I think he weighed 115 pounds in this photo, or a 400-pound sumo wrestler. It turns out that the brain regulates your long-term energy stores in the form of fat homeostatically. And So if you look at the graph, what you see on top is an obese mouse, and the curve on the bottom is a lean mouse. And FR is dieting or food restriction. We can restrict their access to food, so we're dieting them. But when you give them back food ad lib in the middle section there, they come right back up to their old weight. And humans do that as well. That's why it's almost impossible to lose weight and keep it off through diet and exercise. Even the new weight loss drugs will produce very significant weight loss. If you go off the drugs, you put it back on. So we've known since the 50s, from the work of Kennedy and others, that body weight is regulated homeostatically. And so Kennedy's work in the early 50s showed that this was the case and that the hypothalamus was important to that. But we can think about this very simply, actually. Any physiological system that has homeostasis is really no different from the thermostat in your room that keeps temperature, energy in the form of heat, constant. And you really need only a few things to accomplish that. You need a signal. In the case of your house, it's the heat or the cold that the thermostat is sensing. You need a device to sense that signal, the thermostat, and then you need effectors. And that could be a heat pump or an air conditioner that when temperature goes too low, you open a window, temperature goes down, the heater goes on and brings the energy back up to normal. Well, biological energy homeostasis is just like the control of room temperature, as I'm going to show you. Now, when I began my career back in the early 90s, there were no known obesity genes. We knew the hypothalamus was critical for sensing your long-term energy stores in fat, but that's all we knew. We had no molecular or cellular understanding of how this worked. But there were 5 monogenic obesity strains in the mouse, single gene mutations that had been collected at the Jackson Labs in Maine. And the researchers there had saved these and bred these strains because it was shown that a single gene was causing these morbid obesity syndromes. So the mouse, the C57 mouse, is a normal wild type mouse for lab research. And these 2 strains are 2 of the monogenic strains that existed. And they gave them funny names: OB, DB, fatty, tubby, and agouti. OB was obesity, DB stood for diabetes. My work focused on the agouti mouse, but approximately around 1993, Jeff Friedman at the Rockefeller cloned the OB and DB genes and basically identified the signal that fat uses to tell your brain how much energy you have on board. He showed that the OB gene encoded a fat-specific hormone only made in fat that he called leptin. He and others showed that that hormone was made in proportion to how much fat you had on board. He showed that DB encoded the receptor in the brain for leptin. And ultimately it was found that patients with mutations in either OB or DB had the same phenotype as those mutant mice, early onset morbid obesity. And bad news here though, leptin was named leptin because he thought it was the hormone that would make everybody thin. Well, unfortunately, as more research was done, it turned out that the adipostat in your brain, the device that senses fat, can only sense a decrease. As you gradually gain weight, your brain keeps adjusting to the higher levels of leptin, and you don't respond. And so, despite biotech paying an enormous amount for the rights to this hormone and doing a clinical trial, patients who were administered excess leptin essentially lost no weight. So effectively, just like your thermostat can sense a decrease in temperature or an increase in temperature, the adipostat in your brain really only senses a decrease in energy stores. So I came into this field really accidentally. My lab was working, as the technology for cloning genes became available, my lab was working on cloning the genes for receptors, hormone and neuropeptide receptors, and trying to understand how they work just from the basic science of it all. And so we cloned a family of genes that we called the melanocortin receptors that were activated by a family of peptides called the melanocortins. And the first 2 receptors we cloned correlated with known hormone receptors or peptide receptors. The MSH receptor in your melanocytes that regulate hair color, and your ability to tan, and the ACTH receptor in your adrenal cortex that regulates control of the stress hormone glucocorticoids. And so we cloned those two and we characterized them, but we also found 3 orphan receptors that were expressed in different places, and there was no correlation with any known biology at the time, and we very creatively labeled them MC3, 4, and 5 because we had no idea what they were doing. Now, this was a really fun period of my career because after cloning the pigmentary receptor in melanocytes, I got to travel all over the world and collaborate with scientists studying how these genes regulated coat colors in all kinds of domesticated animals. And this is from the Agricultural University in Norway, where I collaborated to discover what makes the red fox red. Ultimately, dermatologists and human geneticists discovered that the same mutation is responsible for red hair in humans. In fact, about 80% of true redheads are homozygous recessive for the MC1 receptor. And there was another gene called agouti that caused a lot of the same sort of coat color variation. Indeed, much of coat color variation in mammals results from MC1 receptor, the agouti gene, and interactions of these genes. And so then I became fixated on trying to understand, okay, if you knock out the MSH receptor in a mouse, you get this skinny yellow mouse, but if you have a mutation called agouti, you get a mouse that's not only now goes from black to yellow, but is obese. And given these orphan melanocortin receptors, it occurred to me that they may somehow be involved. So we proposed that perhaps the agouti gene blocked some of these orphan receptors, and maybe they were involved in determining body weight. And indeed, when Greg Barsh cloned the agouti gene, all that it told us was that it encoded a little protein. But the gene rearrangement itself went from making agouti only in the hair follicle to making agouti all over the mouse. And so we decided to test the hypothesis that agouti might be an endogenous antagonist or blocker of these other melanocortin receptors. And we had shown that MC3 and MC4 in particular were primarily expressed in the brain. This may sound like an obvious experiment, but at the time there were no endogenous antagonists of this family of receptors, and everybody sort of poo-pooed this hypothesis. But it turned out to be true. If you look at the curves where there's the difference between the two curves, that's sort of a classical hallmark of a competitive antagonist. So we showed that agouti, the agouti protein, was a competitive antagonist of the MSH receptor in the hair follicle, but also specifically of the MC4 receptor in the brain. And so it took us, this wasn't as simple as cloning a gene given the complexity of the biology. We really had to do a lot of experiments to convince the field that this was indeed the reality. And so as I said, Greg Barsh showed that the obesity syndrome resulted from this rearrangement of this gene, making this peptide now everywhere instead of just in the skin. We showed that agouti blocked not only the pigmentary receptor, but also this orphan receptor in the brain. We mapped the receptor in the brain and showed that it was in regions of the hypothalamus that were thought to control energy homeostasis. Then the two critical experiments where we deleted the MC4 receptor in mice and recapitulated the obesity found in the agouti obese mouse. And we also developed the first synthetic blocker of the MC4 receptor, and we showed that treating animals with that could stimulate food intake and cause the hyperphagia seen in the obesity models. And literally within a year of that experiment, two different groups, Philippe Frogel in France and Steve O'Reilly in England, identified patients that had mutations in the MC4 receptor, shown in the boy on the right. And Annette Gruters earlier had found the first child with this syndrome involving a mutation in the gene that encodes the neuropeptide, the endogenous neuropeptide that activates the MC4 receptor. In any event, a multitude of syndromes have subsequently been discovered that cause morbid early onset obesity due to mutations in this system. And in fact, mutations in the MC4 receptor shown in this boy here are actually very common. About 1 in 1,000 individuals have morbid obesity early onset due to mutations in the MC4 receptor. And we can model all this in the mouse. So effectively, if you have mutations in leptin, leptin receptor, or the MC4 system, you can't establish a body weight set point because your brain doesn't know what your fat levels are. You're either missing the signal or you're missing the MC4 circuitry in the brain essential for interpreting that signal from your fat. And so rather than maintain homeostasis, you just continue to gain weight. And so this was really validated by about 1997. The first drug to treat these patients was developed by Rhythm Pharmaceuticals in 2020, just to give you an idea of how long it takes to go from a basic discovery of a fundamental physiological mechanism to a treatment. 23 years. If you look at the growth curves here, you can see that these are normal pediatric growth curves, 50th percentile, 97th percentile, 3rd percentile. These are the growth curves of 2 patients with melanocortin obesity syndrome. And then treatment with Imcivree, an MC4 agonist. Basically, it doesn't provide 10 or 15% weight loss like Wegovy and the GLP-1 drugs on the market. It basically normalizes, produces massive weight loss, and really normalizes these children. I said we'd cloned 3 orphan receptors. I won't talk about MC5 today. It's not related to obesity, but we cloned another one called MC3. We mapped it in the brain, and its sites of expression are in yellow, the MC4 sites are in blue, and what we knew from this was it's in different places, and so it must be doing something differently. But there we go. And of course, the tools nowadays for studying these kinds of things are absolutely amazing. This is a mouse brain in 3 dimensions where we've labeled every single MC3 receptor in the brain, every single neuronal cell body, and we're flying through the mouse brain here looking at the location of these, and at the bottom you see a very dense region. This is the arcuate nucleus, a critical site for leptin action. So we knew that MC3 had to be involved. We knew that it had to be involved in energy homeostasis, but the problem was that when we deleted it from the mouse, not much happened. And just in the last year or 2, the first patients have been found that are homozygous recessive for the MC3 receptor. They don't get obesity. But I wasn't willing to give up. The lab wasn't willing to give up. There's a saying in the field from a famous developmental biologist in the UK, you can't say a mutation or a knockout mouse doesn't have a phenotype until you take it to the opera. You can't just look at it under normal conditions. You really have to intensively study it and think about it. And so really, since coming to Michigan, I've been focusing in on the MC3 receptor. So these animals eat normally. So here's an MC4 knockout mouse. This is 24-hour food intake. It eats a lot more than a normal mouse in black. The MC3 mouse eats the normal amount and doesn't really become obese. But the moment you begin perturbing it, then you see some remarkable things. So, oops, I need to go back. There we go. So you see some remarkable things. If you stress the animal, it will eat less than a wild-type animal. So this is simply taking the mouse from group housing, and they're social animals, and putting them in individual housing. They don't eat as much. And then look at this remarkable experiment. Here, we've dieted the MC3 knockout animals in blue versus the wild-type in black. They lose a lot more weight, but then when you give them high-fat diet, they gain more weight. So depending on how you stress them, they're unable to maintain homeostasis properly, but they have a problem in both directions. And so what we proposed is that unlike the MC4 receptor and leptin system, the MC3 receptor has a problem regulating the boundary conditions. It can't sense when you've escaped the boundary of homeostasis, and that's true in either direction. And we've learned a lot about the molecular basis for this, but I'll simply summarize it by saying that the neurons in blue that provide the ligand that activate the MC4 receptor to restrain food intake act to do just that. They release their neuropeptide, it contacts the MC4 receptor. Ultimately, that ligand then contacts the MC3 receptor, which results in the release of the primary inhibitory neurotransmitter, GABA, onto those same neurons and preventing them from further inhibiting food intake. So that's how the boundaries get set up, and you can imagine if you remove those orange neurons, you have unrestrained anorexigenic activity and you see more weight loss. So I'm going to wrap up by trying to address what I'm sure all of you see too much on TV, and that is ads for Wegovy and Zepbound and all these new really miracle drugs that are being used to treat obesity. And they're highly effective on average. People are seeing 15 to 20% weight loss on average for people with normal dietary obesity, not genetic syndromic obesity. But if you actually dig into the results, you see something quite different. And that is, this is a waterfall plot showing the amount of weight each patient on Wegovy loses over 6 months. And in fact, about 15 to as much as 20% of people really don't respond to the drug and don't lose weight. Some people lose a very significant amount of weight. And then there's a high discontinuation rate due to the nausea from these drugs. Now, these drugs act in the brainstem. And this is a region involved in nausea and emesis. These drugs act here, primarily in the brainstem, to make you feel full. And that's how they work. They make you feel full, you eat less, and you lose weight. The melanocortin pathways that respond to energy homeostasis are located deeper in the brain, in the hypothalamus, and in other regions. But in order for the GLP-1 drugs to work, the signals from the brainstem get transmitted to the melanocortin system and other circuits deeper in the brain. And so we asked the question of whether we could harness the melanocortin system, and I showed you the tremendous weight loss that happens in patients with syndromic obesity, if we could harness that to make the GLP-1 drugs better, to either allow physicians to reduce the dosage so that the nausea and other side effects aren't experienced, or to provide additional weight loss where needed. And so these are the results we're seeing. This is a 14-day weight loss experiment in obese mice, and the weight loss from Wegovy is shown in gray. This is a dosing in orange. You can barely see it. It parallels the saline treatment. This is a very low dose of a MC4 agonist in orange. When we add that to the Wegovy treatment, you can see a very significant increase in the Wegovy-induced weight loss. So we think there's going to be a lot of applications of the melanocortin system to fix some of the aspects of these current drugs that are problematic. And so I've been funded by many agencies, including the NIH, the NIDDK, a number of pharmaceutical companies. As I indicated in my conflict of interest statement, I'm working with a company I founded called Courage Therapeutics to try to develop melanocortins for weight loss. And I also want to just mention I have my family here who all live in Ann Arbor now, and they've provided incredible support for me over the years to pursue this career. So I want to thank them for coming today too. Thank you very much.
Thank you for your lecture. We have questions, and we can begin here.
Thank you, Roger. I'm always fascinated by your work. I'm curious, I'm familiar with other G protein-linked 7-transmembrane domain receptors, that they're instances of constitutive activity. Does that happen with MC4?
Yes, it has a lot of constitutive activity.
I mean, are there genetic conditions that have a phenotype with a constitutively active receptor?
There are a couple claims that there are some alleles of the MC4 receptor associated with leanness. And by the way, leanness is, your body mass index is highly heritable. It's very heritable on the high end, and leanness is also highly heritable. So it is probably a leanness. Some of the alleles that increase the activity of the receptor or improve its stability on the plasma membrane probably contribute to constitutive leanness as well.
Thank you.
Question?
That was great, Roger. Thank you very much, and congratulations on the professorship. You mentioned it's hard to escape your set point. You can diet and exercise your way down, and you go back up again. You can drug yourself, you go down, you go back up again. What is it that establishes the set point? What causes the variation in the population? And why does there seem to be a trend towards increasing obesity in society? Is it a genetic trend, or is it more complicated than that?
So a lot of questions there. Great question. And this is a really important question because if you look at any particular given population, you'll see sort of a bell curve of body mass index, which is one of the measures of adiposity that is commonly used. You'll see a bell curve. And the way I like to describe it to people is that, in a lean environment, that whole bell curve shifts to the left. In a highly obesogenic environment, that whole bell curve shifts to the right, and more people become obese. So because of the nature of the adipostat, in an environment in which there's essentially unlimited calories for many people, very little hard labor for many people, there's a natural tendency to become obese. That obesity occurs slowly over time, and the brain doesn't adapt to that and try to get you back to your earlier weight. There are a couple of fascinating studies that were done years and years ago that would never be approved by a review panel today, but the Vermont prisoner study, for example, where inmates were given less than their caloric need to sustain their body weight for a very intensive, short period of time. And likewise, another component where they were overfed very rapidly in a very short period of time. The individuals who were overfed very rapidly went back to their old weight naturally. So we're learning more about the adipostat. It is theoretically possible one day to find a way to tickle the adipostat in just a certain way such that you gain a healthy weight and remain at that weight. But none of our, well, bariatric surgery in a certain percentage of patients accomplishes that, and there's a lot of study going on, but it's not really understood why that's the case. So there were a lot of questions there. I tried to address a few of them.
Great, we'll sneak in one last question in the back, or there, yep. And then unless others will go for at the reception. Please go ahead.
Yeah. Thank you, Dr. Cone, for this great presentation and great research. Great question. I'm wondering what your view is on a different type of treatment by ablating the brain. You might have heard of high-intensity focused ultrasound that can be used to very carefully and with targeted methods ablate the brain to, for example, cure essential tremor. And just recently, it was shown that it can also modulate the desire to overeat. So it has been found that with careful ablation of parts of the brain that you can significantly decrease the desire to overeat, but still maintain a healthy intake of food.
At first blush, without knowing these specific studies, I'm going to guess that that's going to be highly problematic. And the reason why is these circuits I've described to you today regulate a whole host of other functions as well. For example, fertility and pubertal maturation. These mammals have evolved to become fertile and to maintain fertility as long as they have adequate energy stores on board. So the gating signal for puberty, for example, are these very same melanocortin neurons. The gating signal for normal reproductive cycling requires a signal from these same melanocortin neurons, and they're bundled in with whole sets of other neurons regulating different aspects and very different physiological functions based on how central energy homeostasis is to life. So the concept of using some kind of ablation or energy to disrupt just the feeding circuits is probably problematic.
Thank you very much. Please join me in thanking our distinguished university professor.
Thank you, Professor Cone, and I'm sure Tachi Yamada would be so honored to be associated with you in your work. Okay, our final lecturer this evening is Jeffrey A. Fessler, the William L. Root Distinguished University Professor of Electrical Engineering and Computer Science and Professor of Biomedical Engineering in the College of Engineering, as well as Professor of Radiology in the Medical School, a paradigm of interdisciplinarity. Professor Fessler has made profound contributions to the mathematics and algorithms that transform raw data into the images that clinicians and researchers rely on every day. His innovations in computed tomography have enabled lower dose CT scans. This work has made a tremendous impact, as 70 million such scans are performed annually in the United States. His work in MRI has advanced faster, quieter, and more accurate imaging, and his Michigan Image Reconstruction Toolbox has become a global measure for the imaging community. His lecture, and I love the title, "The AI of the Beholder in Medical Imaging," explores how machine learning can improve image formation and interpretation, making medical imaging safer, faster, and more powerful for patients and physicians alike. Please join me in welcoming Professor Jeff Fessler.
Thank you, Laurie. Thank you very much for that introduction, Laurie, and thank you all for being here today. I'd like to add one note about my biographical journey here. For those of you who don't know, when I came to Michigan in 1990, I started as a postdoctoral fellow in the Kresge building that's just a block and a half from here. And I think it speaks a lot to the collaborators that I've had, the students that I've worked with, the supportive environment at home, thank you, Sue, that I get to talk to you as part of this group of distinguished university professors today. When I became the William Root Collegiate Chair about 10 years ago, I gave a presentation where I focused on a retrospective, talking about the work that my group and I had done up until that point, and talking a lot about my collaborators and my mentors. Today I want to talk more about current work and be a little bit more forward-looking. I still greatly appreciate all of the collaborators and my support at home, and I want to just use this one phrase here as a short version of that appreciation. The work that my collaborators and my students and I do is very much under the hood of MRI and CT scanners and other kinds of scanners. To be more precise, it's under the hood of the computers that sit next to those scanners. And it's a little bit technical, and so I thought for this broader audience today that I would try, if I may use an imaging metaphor, to zoom the lens out a bit and talk about medical imaging in the broader context of medicine, and then a little taste of what we do under the hood of these scanners at one point. And I'm going to talk about this broader context in a maybe not so hypothetical example of somebody injuring themselves in a sports activity and then having various medical images along the way, leading eventually to diagnosis and treatment. I'm going to talk about each of those steps. So the first step when a patient presents with some sort of ailment is to decide what kind of diagnostic tests, if any, need to be run, and often those tests involve imaging. But even once you decide you want to make images, there's lots of choices there. There's so many different kinds of imaging methods, X-ray, CT, MRI, et cetera. And even if you decide you need an X-ray because you need to know whether that shoulder bone is broken or not, there's choices. Do you inject a contrast dye or not, or do it without contrast? And so there is a field adjacent to mine called AI-driven clinical decision support systems, where people are taking the patient's history and the medical record and other characteristics and deciding what diagnostic tests should be made for a given set of symptoms. For something like a dislocated shoulder, it's very obvious you need an X-ray to find out whether the bones are broken. But if you have a patient show up with a headache or dizzy spells, it's much less obvious, perhaps, what kind of diagnostic tests are needed, and perhaps AI methods could be helpful in that setting. Now, X-ray images are very useful for showing bones and seeing whether they're broken or not, but they're not very good for seeing subtle soft tissue structures like whether or not a rotator cuff is torn or not. So a specialist like an orthopedist is likely to order a kind of scan that is more capable of showing soft tissue contrast, and magnetic resonance imaging excels at showing soft tissues. But an MRI is actually extremely complicated, very versatile. MRI scanners typically have hundreds, if not more, protocols, different ways you can image. Each protocol has dozens of selectable parameters. The obvious ones are like, do you image this way or that way or whatever? And then there's control over the number of slices, the pixel size, the type of contrast you want. So you would not be surprised at this point. You can see where this is going, is that there is also adjacent work adjacent to my field of AI-based protocoling, based on the patient's history, patient's conditions. What are the appropriate protocols to select from the MRI scanner? After you take the scan, you produce some images, and then it's the job of a radiologist to read those images and make a diagnosis. And I have incredible respect and admiration for the radiologists that do this job. I'm showing you 3 example images here of a shoulder, but in reality, typically they are reading stacks of 2D images. They're scrolling through them. If you've never seen it, it's not unlike what you might have done as a kid with a flip book, looking at lots of 2D images to try and make sort of a 3D mental picture of something going on. It's amazing to me how they do this. This is the part where somebody who didn't come to the talk is probably thinking this is where I'm talking about, right? The term AI and the beholder, this is the most obvious place that you might think about applying machine learning or AI type methods, because we have this whole field of computer vision that tells you whether this picture contains a cat or a dog, and that same technology can be used to decide whether this picture has a tumor or not in it, right? And so there's a whole field, even closer to mine now, on AI-based image classification, image analysis, taking medical images and processing them to try extract information, diagnostic information from those images. This is such a perhaps obvious application of computer vision type technology that in 2016, Geoffrey Hinton, who's known as the godfather of AI, said people should stop training radiologists now. It's just completely obvious that within 5 years, deep learning is going to do better than radiologists. That was 2016. We're in 2025. By the way, he got a Nobel Prize in physics for 2024, maybe not for his predictive abilities, but for his very strong impact on the field of machine learning and artificial intelligence. I don't see personally that computer vision techniques are going to replace radiologists anytime soon, but I think it's likely that they will augment radiologists and assist them in the diagnostic process. One of the reasons I think it's non-trivial, I should have mentioned on the previous slide, is that most computer vision technology is designed for processing 2D images, right? Because we all take pictures with our cell phones. There's lots and lots of such pictures online, lots of opportunity to train machine learning methods with those pictures, but those are just 2D pictures, and that's quite different than the stacks of 2D images that really give us 3D volumetric imaging. That's more challenging to, but it's an active field to work on using machine learning for making the diagnosis. And just to tell you how active it is, there's a survey paper that just came out in Nature Digital Medicine this summer that looked at over 1,000 FDA-approved, FDA-authorized applications corresponding to over 700 devices. And you can see here that about 600 some of those is imaging, and then a few are signals, heart waveforms, brain waveforms, and so on, being used for applications like image analysis and image generation. So this is clearly, I don't know what fraction of those 700 unique devices are actually deployed in the field, but I'm sure many of them are, since companies have gone to the trouble of getting those authorizations. So this is beyond research and development. This is happening around us, and I'll give you a concrete example of that in a few minutes. Finally, after you make the diagnosis, you need to make a treatment plan, and this is another adjacent area of AI, is using AI for treatment planning and monitoring. The treatment here could be pharmaceuticals, like we talked about a minute ago. It could be radio-labeled antibodies, like some of my collaborators use, or radiation with external radiation sources, like others of my collaborators do. There's just lots of different aspects of designing that treatment, and you could imagine that AI could be helpful in that aspect of medical imaging as well. All right, so that's the big picture. Now I'm going to zoom into more of what my collaborators and my group and I work on, the under-the-hood part of it. This cutaway view of an MRI scanner shows the 2 parts that we engineers and physicists have the most control over, and that is the radio frequency coil and the gradient coils. The radio frequency coil is what sends signals into the body, and then it also senses the magnetization that's produced in the body as a result of that excitation, as we call it. And then there's the gradient coils that we also can control, the signals that go into those coils. And for the purpose of this discussion, that's what controls the spatial resolution of the scanner. All right, I'm going to tell you a little about what we do, but first of all, let me tell you why we do it, and Lori already alluded to this in her introduction. So one of the thing I'll focus on the most is that we're doing this to try to reduce scan time. Reducing scan time makes it more comfortable for patients. Nobody wants to lay down in those tubes for a long, long time, and obviously that's more cost efficient because you can have more patients per hour through the scanner. And if the patient is in the scanner for a shorter period of time, they're more likely to hold still. And if they move around, you're going to get motion artifacts in the image. So it's kind of a win-win to make scans shorter. We're also trying to make them quieter, trying to improve the image quality, better spatial resolution, lower noise, as we call it. In many of my collaborations, we're trying to make the images more quantitative instead of just giving you a subjective impression. They're trying to make actually quantitative values, trying to make those values reproducible across different scanners from the same company, but different scanners from the different companies across sites and so on, so that you can pool data from different sites for group studies of patient statistics. And then finally, we're trying to make the images more specifically related to tissue pathology. And we have some studies in white matter related to that ongoing right now. Okay, to go a little bit deeper, I have to tell you just a little bit more about how the data is collected in an MRI scanner. When you take a picture with your cell phone or a digital camera, the light comes through the lens, and it's recorded on a 2D sensor array, and the numbers that come out of that array, you could view them directly on a computer screen, and the numbers would be interpretable to the human eye. The companies, they do some processing of those raw numbers to make the image look a little better, and we've had a lot of images today. There's a recurring theme of imaging in these talks. But it's quite different in an MRI, CT, or ultrascanner. The raw data that comes out of those systems looks something like the image on the left here that's uninterpretable by a human. It has to be put through very complicated algorithms, I've summarized with a few equations here in this gray box, to produce the reconstructed images that are finally viewed by the doctors to make their diagnoses. Now, the way we collect this data, traditionally in MRI, this case-based data it's called, for those of you who know, this is the two-dimensional Fourier transform of that slice of that image there. We collect it by manipulating the gradient coil waveforms to sweep one row at a time, top to bottom, left to right, kind of like you read a book in Western culture. And this is why MRI scans are slow. It takes a certain amount of time to read one line at a time like that. So there's been lots of research on trying to speed it up, and all of these diagrams I'm showing here are other ways of collecting the data. The ones that you see more gaps in, those are designed to take less time. If you don't read every row, it's kind of like skimming a book, I guess, speed reading or something. It takes less time. You have less data now, and so you need more sophisticated algorithms to produce a decent image when you collect less data. These designs here are all what we would today call handcrafted. They're based on engineers and physicists' intuition about what might be reasonable ways to collect the data that hopefully can produce good images. But they're not really based on the statistical properties of any given group of patients. So one project that we've been working on recently is trying to use statistics, and this is where the machine learning comes in. So if you allow me to walk through this block diagram, what we do is take a bunch of high-quality MRI images, shown in the upper left there. We propagate them through a mathematical model for the physics of the MRI scanner to compute, actually, simulated case-space data. We put that simulated case-based data through a sophisticated image reconstruction algorithm that includes convolutional neural networks, or deep learning, AI if you want, approaches to produce reconstructed images. The images that come out initially will not agree very well with the images that we started with, and so then we use an optimization technique to update the sampling pattern in case-space and the parameters of the image reconstruction method over and over until we get good images that come out. And so this is called joint optimization of the image reconstruction method and the case-space trajectories. After we've done that offline optimization process, we take the learned, our optimized sampling pattern, we program that in the MRI scanner, we collect new data, and then we put it through the same optimized reconstruction method to get the final output image. All right, so we need a library of images to do this, and there are thankfully a growing number of libraries of images. And one thing we learned from this process, before I go on, notice I didn't say the word AI anywhere on this slide. Okay, I don't view any of this as intelligence. This is machine learning. We're teaching a machine how to do this process using training examples. But the field has gone for the marketing of AI, so I just want to draw that distinction here. So we have trained this separately on batches of knee images and batches of brain images, and what we find is the sampling patterns that are best from this machine learning process are different for knees and brains, which makes sense. The statistical properties of knee images and brain images are different from each other. So that's kind of interesting. And we take one of those, the brain pattern there, we program it in the scanner prospectively, and we actually have some results here. I don't know if you'll be able to see in the screen, but the traditional method of undersampling on the left produces some artifacts that are cleaned up by my former graduate students' optimized method. So just one real example. Now, that work I just described there is optimizing the case-based sampling for a group of patients, a cohort of patients. The holy grail in this field would be to optimize the pattern for every individual patient, very patient-specific medicine, and we have some very preliminary results. This is much further from being deployed in the field. And so just at a high level, we're also working on trying to take a little bit about data of a patient and then saying, okay, what other data we need to get the best possible image for this patient? We call that patient-specific case-based sampling. Now, no talk on AI would be complete these days without mentioning generative AI. So in another collaboration with a student and colleagues, we are trying to learn the statistics, in this case of CT images, so that we can, well, we want to learn those statistics so we can do a better job with image reconstruction. So what's shown in this slide is 3 different generative AI models. These are all, you could call them deep fake CT images. The top row is a method that takes a lot of training data. The middle row is a method that uses patches that takes less training data, but as you can see, leads to these sort of jigsaw puzzle-like images. And the bottom row is the one that we're promoting that takes a modest amount of training data, produces not too bad looking CT images. Now, there's really no scientific use in generating by itself deep fake CT images, but the reason we do this is a clue to us that we've done a good job of learning the statistics of CT images. If we hadn't done a good job of learning those statistics, we wouldn't be able to generate images that, at least to the layperson, look somewhat like CT images. To a radiologist, you'll see some parts that are not quite in the right place. But we're generally learning the statistics. Once we have those statistics, we can do a kind of image reconstruction algorithm that takes lower dose, so therefore safer for the patient, less X-ray intensity. And we're doing it here by what's called sparse view CT, collecting fewer images, basically, and then reconstructing good quality images because we're exploiting the statistics that we've learned. I'm being very superficial in the details here. If you want to learn more, on October 28th, I happen to be giving a seminar in the statistics department where I will give the gory details. All right, and a talk on AI would also not be complete if we didn't talk about some of the caveats and issues with AI, all right? So there's typically very large data requirements, which we have in some settings, but not in all medical imaging settings, so we're working on that. There's privacy issues, so there's a whole field related to try and train these models while preserving privacy. The CO2 footprint has to be thought about, and so on and so forth. But this is not just research. It's already there in hospitals. I happened to have an MRI scan of my shoulder a couple of weeks ago, and the MRI technologist who was setting me up in the machine said, "You are so lucky because just a week ago we got a software upgrade and your scan is going to be 10 minutes instead of the usual 25 minutes because of the new AI feature, the AI upgrade." And I've been in the MRI scanner many times because we do it all the time for research, so I am totally comfortable laying on an MRI scanner, but still, I'm perfectly happy to get out of there in 10 minutes instead of 25 minutes. And for people who are claustrophobic, this is big. Right? So this is not just hypothetical research, and I'm not saying I'm personally responsible. This is the field that I work in has made this advance, and that's quite a significant to say this is a 10-minute scan now instead of a 25-minute scan. All the images that I've shown you here today at the end, those are results from my groups, from my current and recent group members, and some of you know that I have a musical hobby, so I'd like to end on a musical note, pun intended, which is, it is very true and honest that as a teacher I have learned so much from my pupils. Thank you very much for your attention.
Thank you very much. We can have questions for our lecturer. In the back, and then in the front here, next.
Congratulations, Chef. This was great. Thank you so much. Quick question. Is this a new sampling method for the case space, is it lossless, so to say? Because let's assume it works on average for a great population, that's wonderful. But if I have a small defect that would only be seen in fully sampled case space, I would fall through the crack.
Yeah, that's a great question. I don't know if I can go back or not. I sat on an NIH study section once where the medical physicist who was the chairing that study station, we were reviewing proposals about compressed sensing MRI, where you collect less data. Your question's about, is there risks associated with collecting less data? And I remember that chair of the study section said, if my kid's in an MRI scanner, I want every point in case space sampled. And I thought, well, that is great as long as your kid is willing to hold perfectly still, right? Or do you want to sedate your kid, right? So anytime we undersample, there's the risk of losing information. Now, one thing I didn't mention here, is not shown in the cutaway diagram, is these scanners all have multiple coils. So if we only had one coil in the scanner, it would be quite risky, I think, to undersample case space by the factors we're talking here. But the scanners that we have on North Campus have 32 and 64 receive coils. So there's a lot of extra information from the multiple receive coils. But still, your question is an excellent one. We don't know yet how much faster we can make the scan without possibly losing important information. Apparently, the Siemens company that made the scanner that I laid in for 10 minutes recently has convinced the FDA that they can reduce it from 25 to 10 minutes and it's still adequately safe. I have a former student that works at the FDA. I'd like to actually have a conversation with her someday and find out how exactly are they confident. And I'm not trying to cast any doubts here, but I think it's very complicated to know whether or not the AI that's in here is going to work every time or not, okay? I think that's plenty of opportunity for open research, but apparently it's working well enough that the FDA had enough confidence, and this was approved, let's say, before January 2025, to approve that technology, okay? I hope that kind of answers your question. But I think that's a really important research area, is to understand better the potential for hallucinations of either positive or negative outcomes.
Great. Here and then here.
So kind of to follow up on that discussion. So I don't know how true to life those trajectories you showed for knees and brains are, but I'm wondering in terms of actually implementing these sampling strategies. So all the classical ones have some sort of regularity to them.
Yes.
And if things get kind of more irregular and nonlinear, can you actually maintain the fidelity of the procedure?
So great question. So in practice, what we do, the scanner is not perfect. Even for the regular trajectory, sometimes we have to make corrections. The ones that I showed you, that were radial or spiral especially. So in fact, the student with my collaborator, Doug Knoll, in the back of the room, who did this work, had to actually program the scanner to play that trajectory and then use a special kind of calibration process to measure what did the scanner actually do, because it doesn't do exactly what we want it to do. And then we use that so-called measured trajectory in the image reconstruction process. So it's not quite as simple as plug and play, but there's well-established techniques for compensating for the imperfections of the scanner. So very insightful observation.
Isn't it? I'm Paul Carson.
Hi, Paul.
Medical physics.
Paul also used to work in the basement of the Kresge building a block and a half away, just like I did. Anyway, go ahead, Paul.
Isn't it possible to do MR scans with both sets of case space sampling, the full set and your set, and then compare the results to see what you lost?
Yes, as long as the patient held still. And so, yes, on the slide I showed, I don't know, in the paper, I don't know if it was on the slide I showed, we have the fully sampled data as reference, and then we're comparing it to the undersampled data. So absolutely, all of the papers are full of, PSNR measures, mean squared error measures, and so on to compare the reconstruction from the accelerated scan to the fully sampled scan. That works for things like the brain that hold relatively still. It's more complicated for things like cardiac imaging or dynamic imaging where you're introducing a contrast agent. Then we can't really collect both the fully sampled data and the other sampled data to compare because things are changing in time.
Can you approximate the new sampling from the fully sampled?
Well, I would say, some of my collaborators have heard me say, I believe there's no such thing as fully sampled dynamic data because the object is changing as you're collecting the data. There's a lot of things we call fully sampled, but it's not really fully sampled. But to the extent you're willing to make that approximation, yes, we can do what you described. But it's never perfect because of the dynamics.
Last question in the back there.
As you, Rex.
It'll take a minute for a microphone, but if you could wait, be grateful. Thank you.
Thanks for the wonderful talk, Jeff. I was wondering, you mentioned that much of the current state-of-the-art AI tools only are trained on 2D data. So what do you think it will take for AI to work with like 3D data or even 3D time series data?
It's funny because just a couple hours ago I had a meeting with one of my collaborators and one of our students, and we were working on models for 3D CT scans. So it's an active research area. The challenges are both the amounts of training data, because 3D volumes are bigger, and the GPU memory. And it takes more memory, right? And the GPUs, the graphic, the NVIDIA cards that you all hear about in the newspaper that are used to train these algorithms have a limited amount of memory, so the number of 3D scans or patches of them that you can fit in is limited. So I think we need, to answer your question, we need smarter algorithms and probably continued advances in the computing hardware so that we can move to 3D and then eventually, like you said, 3D plus time, which is what many of us here are really interested in MRI. Great final question. Thank you.
Great. Thanks, Jeff. Let's thank our speaker. To conclude our event, I would like to thank our three distinguished university professors and invite you all to a reception beginning now, just through the doors on my left. Thank you very much for joining us, and please have a good afternoon.